Well, hello, and we are now doing Bio 211, AMP1. We're looking at Chapter 14, and we're looking at the brain and the cranial nerves. For those people who are not at Norwalk Community College, these slides have been adopted or adapted from the text that's used inside that class, and that is Tortora and Derrickson. We're beginning at the brain and the spinal cord in development, and what we're looking here is the 3 to 4 week embryo. Before I get going, though, I just always feel like I have to say this because, unfortunately, I feel like some students are just simply going to watch these videos and maybe the Khan Academy things that I've curated as well, and they're not going to read the textbook. To fully prepare for this class, it's expected that you've read the textbook either before or after you've watched these or you've gone to lecture or something like that. But the only way you can get all the bits and pieces to put the whole story together, no matter how many details I try to put into these presentations, it's not going to help unless you've read. So, having said that, we're going to pick up here right around the 5-week embryo, because what's going to have happened is you'll have developed enough to actually have a telencephalon, which will become the cerebrum. Okay, or the diencephalon, which is the thalamus, the hypothalamus, and the epithalamus. And you remember inside class, I've pointed out to you that for essentially most of the regulation in 13 and 16 travels through the thalamus. ANP2 is regulated essentially all through the hypothalamus, and the epithalamus we discuss in both ANP1 and ANP2. The midbrain is derived from the mesencephalon, and the mesencephalon gives rise to the pons and the cerebellum, as well as the myelencephalon giving rise to the medulla oblongata. And we've seen all of these inside labs. So what you haven't seen, though, is really what's actually going on in the development of the brain. All those structures I was telling you about, these are the physical structures. And as they grow, literally what's happening is ends up being the gray matter and some of the white matter inside the space. And the literal cavities between them are where cerebral spinal fluid will travel through the inside and cerebral spinal fluid will travel on the outside. I'm not sure what closed caption is going to do with that pronunciation, but cerebral spinal fluid is going to be the material we're going to make that's going to ensure that all of the cells get the glucose and oxygen and remove the carbon dioxide and the waste that needs to come out of metabolically active cells. Okay, so this is actually how we end up building the cortices or the cortex, as well as the cavities, the space where fluid's going to pass. What are the major parts of the brain? Well, the brainstem is a continuation of that spinal cord, chapter 13. If you haven't watched those, obviously read the textbook first there and then go back and watch those or listen to a lecture and read or something. But that brainstem is that medulla, pons, and midbrain, which is going to give adjacent structures like the cerebellum, which is going to be required for the coordination, actually the intention and the coordination of movement inside the body. You'll see how that's really important inside chapter 16. The diencephalon, as I mentioned a moment ago, has incredibly important responsibilities for making sure that the way station, meaning all sensory inputs that travel into the body through the somatic, I'll make sure you get that here, somatic nervous system, travel through the thalamus to then send it to specific locations inside the cortex. The hypothalamus is going to regulate normal homeostasis, so let's just S-T-A-S-I-S. Okay, and the epithalamus, for the purposes of this class, we're going to think of it in terms of sleep regulation. Sleep regulation. Okay, pretty much that's going to keep you in good stead. If you understand these basic parts of the diencephalon, there are 3 responsibilities. The thalamus for somatic transmission of information to the cortex, the hypothalamus sensing and responding to changes in needs of homeostasis, and then the epithalamus, how it actually is responsible for sleep regu. Let's see if I can spell here. Sorry about that. Regu, L-A-T-I-O-N. Okay. The cerebrum, that's that gray matter where you're actually doing all of the processing. And remember, the cerebrum is going to be where you find interneurons, dendrites of cell bodies of neurons that will actually exit, as well as the axon terminals that have traveled up to that space. And let's see if you can put all of that together as we go along. And this is what I've been drawing since probably the first day of class. Here's a really quick cartoon, and if I pull out the pen and just go like this, just trace the cartoon around, right? Remember, this is what I've been drawing inside class. Boom, looks something like this. I put this thing inside there, and I'll come over here and I'll put that there, and lo and behold, how long has that taken me? Taken me no time at all, right? And what I want to make sure you understand, having done that, is that cerebrum, of course, is the cortex we were just speaking about. And here's where we find the central thalamic mass of the thalamus. And this box right here I'm drawing around right there is making up that diencephalon. Here's the cerebrum right there. Here's the medulla. There's the pons. And here's the midbrain. No different than the spinal cord, I need you to understand that we protect the coverings of the brain with the meninges. And similar to the spinal cord, there is a dura mater, an arachnoid mater, and a pia mater. However, the brain, take a look at the covering here. I'm just drawing a series of covers that are surrounding this in terms of like yellow dots. I think they're following you around here, right before we get to the spinal cord. They're going to be built in a slightly different manner. And I want you to see how, okay? So first and foremost, I want you to understand that the dura mater actually separates into different parts to separate the hemispheres of the brain. And not only do you actually have a right and left hemisphere, you also have the cerebellum you have to separate from the cortex of the brain. So to do this, we are going to use the falx cerebri first to separate the two cerebellular hemispheres. Then the falx cerebelli, notice the difference in spelling here, to separate the two cerebellular hemispheres. And then the tentorium cerebelli to separate the cerebrum from the cerebellum itself. And what this actually ends up looking like, here's one of the cartoons or images you're going to find inside your textbook. You actually have the dura mater, which actually has within it a osteogenic portion and a tissue portion. And what that means, of course, is it's going to allow for space to be filled just deep to the bones that make up the skull. Interestingly, and probably more importantly, is that the dura mater creates the space by which we create sinuses inside of that tissue. Notice where they're traveling. You have the superior sagittal sinus, which is traveling in between the hemispheres of the brain. You have a transverse sinus, which is going to collect blood as it drains from the straight sinus as well as the inferior sagittal sinus. And the whole point, of course, is going to be this. The circulatory system, returning deoxygenated blood, utilizes the two layers of the dura mater to ensure that it's returned back now into the circulatory system. So how does this work? Well, the blood flow to the brain immediately gets, I think I've told some of you inside this class, but not necessarily everybody yet, 20% off the top for all blood supply. And what I mean by that is essentially all the oxygen, all the glucose, 20% of it goes to the brain immediately. No ifs, ands, or buts about it. It's traveling through the internal carotid and the vertebral arteries to carry blood to the brain, and utilizes the internal jugular veins to return blood from the brain. And what's going to happen is we build these blood-brain barriers that we saw back in chapter 12. Let me write that in there for you. Chapter 12. Let me see if I can put that in there. Where we used ependymal cells. The ependymal cells are going to filter material that comes from the capillary beds at the pia mater, traveling to choroid plexuses. You'll see how that happens in a moment. So we actually make cerebral spinal fluid. Okay? So how's that going to happen? Well, it's a clear fluid, yay. And the reason why I'm stating that is, should somebody do a tap, or you do a tap in your future professional life, and that fluid comes out not clear, that is a major identifier for something bad inside the central nervous system. That clear fluid is supposed to circulate through the cavities in the brain. You'll see those cavities are those ventricles we saw inside lab. Remember, we cut the brain into multiple pieces, right? And we saw that there were spaces, and those spaces are ventricles, as well as the spinal cord, that central canal, as well as the subarachnoid space. I shouldn't have to explain to you what the subarachnoid space is, but just deep to the arachnoid matter will be the space where the cerebral spinal fluid would be found. It actually is responsible for absorbing shock, but it also protects the spinal cord. But obviously, it's going to transport nutrients and waste from the blood and the nervous tissue itself. So how does it do this? You're going to see here that this is a rough circulatory system. Don't worry about this stuff over here. Read it when you get a chance, right? But take a look what's going to happen. We are going to make, let's say we're going to use these choroid plexuses, which are going to have ependymal cells that have purified plasma and interstitial fluid of blood through ependymal cells to make cerebral spinal fluid, and they travel through the lateral ventricles, through the third ventricle. Notice we actually have multiple locations where the choroid plexuses can be found, such that we actually are traveling now deep to the subarachnoid space. And then ultimately, inside the dural venous sinuses, the arachnoid villi will collect that waste material into venous blood, send it back to the heart and lungs via the internal jugular vein, and this continues along. Okay, and we just keep going, circle after circle, to ensure that we actually have that formation and circulation of cerebral spinal fluid. You can see it here. So here are these choroid plexuses, and in your dissection I had you go look for these. Remember I pointed out those deep dark brown structures inside all of your dissections. And you have them in multiple locations, and they travel around the spinal cord. They travel through the spinal cord as well, through that central canal. But notice when we drain it back in, we drain it into those subdural sinuses. So this is a good place to stop, because what I'm going to do now is, once we've figured out we actually have a circulatory system, I'm going to transition into the major physical structures of the brain. These are going to say pretty much where we are inside the brain. We're going to start at the lower portions. We'll start down here inside the medulla, and we'll work our way up to the higher cortical functions. Okay.